we all have our genetic background, and we've learned so much about risk variants that modify the risk of developing Alzheimer's disease over the past decade, plus the lifestyle factors and our environment. And I think much of what drives that disease may be coming from gene-environment interactions we have to learn from. So I think if we start to look at large data and mine bigger and bigger data sets about the interaction of our genetic underpinnings and our lifestyle behaviors, we may find hints how to personally advise individuals to change different things in order to have even a better prevention.
Hello, everyone, and welcome back to the Neuroscience and Beyond podcast. I'm your host, Willem, and together with my amazing team, we're on a mission to make science, especially neuroscience, more accessible and exciting for everyone. In this episode, I talked to Professor Michael Heneka, director of the Luxembourg Centre for Systems Biomedicine and one of the leading voices in neuroinflammation research. His pioneering work explores how the brain's immune system contributes to Alzheimer's disease. We talk about why Alzheimer's is such a complex and challenging condition to treat, how inflammation shapes the progression of neurodegeneration, and even explore the surprising world of tunneling nanotubes, tiny bridges that allow cells to share signals and materials. Our conversation shows how these discoveries are changing the way we think about brain health and disease. If you enjoy the podcast, hit subscribe, leave a comment, and share this episode with someone who loves science. And now, dear listeners, please enjoy the conversation with Professor Michael Heneka. Professor Heneka, thank you for being with us here today.
My pleasure.
Yeah. So we have the Horizons conference going on, and we were so lucky to have you as a guest on our podcast. We don't have that much time, but we have a very interesting topic to discuss, and this is Alzheimer's disease and neuroinflammation. Let me start by asking you, what is actually the focus of your research?
Well, the focus of the research in lab is the interaction between neuronal processes, basic mechanisms of neuronal function, neurodegeneration, and the immune system.
These are very interesting terms that you mentioned. Immune system of the brain, you have neuronal processes. Before we go and tackle the Alzheimer's disease and neuroinflammation, can we maybe define these two terms, if you wish? What does Alzheimer's disease mean? And what does neuroinflammation mean?
So Alzheimer's disease, from a clinical perspective, is defined as, dementia characterized by memory deficits, progressive memory deficits, and impairment of cognitive domains, including speech, calculation, visual construction, orientation, and so on and so forth. And the combination most frequently starts with short-term memory deficits and then proceeds into other areas, follows the spread of pathology in the brain, and gives a very distinct clinical picture. From a neurobiological aspect, there are three major pathological hallmarks, which is the extracellular deposition of beta-amyloid peptides in so-called amyloid beta deposits or neuritic plaques. Then there is the intraneuronal accumulation of tau protein, a hyperphosphorylated form of tau protein, which forms then neurofibrillary tangles and accumulates in the neurons. And ultimately, the third component is what we can now maybe summarize under neuroinflammation, which is an immunological response. Initially driven by local and resident innate immune cells, the microglia cells, but also involving other cells in the brain like astrocytes, endothelial cells, oligodendrocytes, and maybe at some stage cells from the peripheral immune system.
Mm-hmm. So everything is involved: neurons, this so-called glia that you mentioned, astrocyte, microglia, oligodendrocyte, as well as blood vessels and cells around them, the endothelial cells, for example. And this is a very complex disease, and it affects a lot of people, not only the people, but also their relatives, friends are also affected by this kind of disease. Do we know when does it occur? You know, we have really difficulties diagnosing it. And is there any point, any way that we kind of detect it at earlier stages of the disease?
So I think once the full clinical picture is there, we do not have a problem really diagnosing Alzheimer's disease. We have, various means by which we can do that. We have neuropsychological test batteries. We have the analysis of cerebrospinal fluid and now plasma markers. We have MRI, and so we can really, with a high security, nowadays diagnose Alzheimer's disease. It's more important to acknowledge the fact that the clinical appearance of the disease is rather in a late stage, and the pathobiochemical processes in the brain start many years, if not decades, before that. At a moment, patients are not patients, patients are population. A patient, by definition, is someone who seeks a doctor's advice for diagnosis, treatment, and similar, but these people do not know that they have an ongoing pathological process in the brain because they don't feel a deficit yet. Mm-hmm. And it will be important to identify biomarkers who are able to indicate who is at risk or who is actually on the way of, advancing to the stage where the clinical symptoms then are becoming apparent. And we have very recently identified a biomarker which can be drawn from blood and investigated, which is tau, phospho-tau 217, which is a marker that may be indicative of ongoing disease 10 years or even earlier before the symptoms appear. So that now enables us to hopefully look at larger numbers of population and screen for people at risk and then treat these people in time.
Mm-hmm. Okay, let's say that this kind of biomarker probably works. I'm not familiar with that. I mean, you can correct me here, of course. And we have a patient or a person that comes to the clinic, and there we detect this kind of accumulation of this tau protein, phosphorylated tau. Can we actually do something to prevent the progression of the disease or to cure it completely?
Well, I don't think we have a complete cure, and that would be great news to everyone. But we have quite a number of measures in the prevention arena, so to say. Mm-hmm. There has been a seminal paper of the so-called Lancet Commission on AD prevention published in summer 2024 by Livingston and colleagues, detailing 14 factors which contribute to AD risk, which are all addressable. And if you would collectively address all these factors through one life, you would reduce the patient numbers in 2050 by about 47%. So we could basically cut patient numbers in half if we would all adhere to these measures and all correct these. There are various things like obesity, hearing loss, hypertension, but other factors as well. Mm-hmm. So reduced physical activity, social isolation is a factor. So many factors at different stages of life. Education is a very early factor. Social isolation is a late factor. Obesity is a factor in midlife. And you see how these various single factors add to an overall number of causing 47%. And I think if we would be able to address all these, we would have a very strong prevention. Imagine we would reduce our patients by half. Now, in the terms of treatment, we have the advent now also in Europe by the anti-amyloid beta vaccination, passive vaccination, which is effective. I would say it's the first stage of a disease-modifying drug. They're not perfect. They're not doing a great job. Mm-hmm. We all know that. Mm-hmm. And I think maybe they can't because they actually affect a mechanism which takes place very early during the disease stage. So I think we will have, over the next couple of years, we will have more and more drugs which look at different mechanisms and which will be used in an additive or synergistic manner.
So if we take, for example, cancer, there are now quite a few therapies that try to target particular cancer type of particular person. Do you think that personalized medicine in treatment of Alzheimer would be a meaningful thing, or is this we are not there yet to know like the specificity of particular person and what is this phenotype, what is it, how is it different than others? Is this possible? Is this meaningful? What do you think?
I like the comparison to oncology, because it tells you that the oncologists have a big advantage because they have something that grows, that metastasizes. They can take samples and probes from. It's very easy for them to do a personalized medicine. If a patient comes to me and reports memory deficits, he usually has a hole where previously the hippocampus have been, and I should explain why there is a hole. But I have no tissue. So it's the inverse problem, and I think that's why the scientists in the neurodegeneration area have a much bigger challenge, at least from that perspective. In the case of how to personalize medicine, we all have our genetic background, and we've learned so much about risk variants that modify the risk of developing Alzheimer's disease over the past decade, plus the lifestyle factors and our environment. And I think much of what drives that disease may be coming from gene-environment interactions we have to learn from. So I think if we start to look at large data and mine bigger and bigger data sets about the interaction of our genetic underpinnings and our lifestyle behaviors, we may find hints how to personally advise individuals to change different things in order to have even a better prevention. If that enables us to also develop very specific therapies, I'm not yet sure. I tell you why. So I'm exposed to this discussion about mining the GWAS information and using this risk variance to indicate and work on pathways and try to therapeutically engage them and use them coming out of this risk variance. TREM2 is a classic in my research field. And I'm not quite sure if it will be always the genetic hint which helps us. We know about the genes responsible for hair loss since decades, but yet if I look in most audiences, there has not been yet a big discovery that one of these genes led to the therapy of male hair loss. So I'm not always sure if the gene hint is the right one. We may have other mechanisms which are more fundamental, where we can interfere with in order to achieve, disease modification, delay, or even protection.
Yeah, there are even also some other diseases that we particularly know the one gene that causes this mutation. For example, Huntington. There's this, but still we cannot, even though we have CRISPR, these tools that can really precisely edit out genes and make things work better in a way, at least in vitro. We still don't test these tools in humans, at least in Europe and the USA. In China, as we know, there were some cases. Although there are some therapies, I think, in vision restoration that already use CRISPR and also AAVs, but this is totally another topic. Speaking of genes, what is the portion or the percentage of people who have this kind of genetic mutation background and then get Alzheimer's? As long as I remember, this is around 5% of all the cases, or I'm wrong?
So you have to distinguish the familiar form and the autosomal dominant inherited mutations, which only involve 3 genes, the amyloid precursor protein, presenilin 1 and presenilin 2. And this is depending on what population you look. It's around 5%, 3 to maybe 6 or 7, depending on precise numbers. But, collectively 5%. And then you have, 15% of the population that carries an ApoE4 allele, which is a very strong risk, and there is even 3 to 4% who are homozygous for ApoE4. And from these ApoE4 homozygous carriers, we assume that when they reach the age of 75 years, 100% of this population will be Alzheimer sufferers. So I think from ApoE4 we know this strong impact. From the GWAS identified risk variants, we know that they also modulate the effect. Overall, the majority of Alzheimer's disease may have a genetic connection. The question is if what else you have to do that this risk is then actually being translated into a real disease.
Yeah, is it a stress? Is it a situation that is... There was recently an article, I think, in one of the big journals, connecting lithium deficiency to Alzheimer's. I don't know if you stumbled upon this one, but it was very interesting that some endogenous lithium in our body kind of has impact also in neuroinflammation as well.
And very low lithium doses would actually help memory formation. That's very interesting. There were a little before that lithium paper in Nature was a paper that showed that if you would get immune vaccinated against varicella zoster, you would have a 20% reduction in AD risk. And so that tells you that at some stage varicella zoster infection might be interfering or a driving force, at least for a fraction of the patient population, and that directly involves a neuroinflammation, because the response system to viral infection, even latent viral infection, would be the innate and adaptive immune response.
That's a great point, actually, to switch from Alzheimer's to neuroinflammation, although they are related. Maybe you can tell us how are they related, how exactly these beautiful microglia cells contribute or kind of try to prevent Alzheimer's disease progression.
The myeloid cells, including microglia cells, have been, developed by nature to defend the body from pathogen invasion. We have to acknowledge that, not very long ago, it was more important to be immunologically resistant and strong as not becoming demented. So the nature rather kind of equipped us with genes and systems to defend our body from bacteria, viruses, fungi, and parasites, which we're constantly exposed to. And so these very interesting work actually shows from Matthew Chapman's lab, published in Science over 2 decades ago. It showed that bacteria, very common to us, including Escherichia coli, Staph aureus, Salmonella typhimurium, carry beta sheet structured amyloids on their surface. Now those are similar to the beta sheet structured amyloid depositing in the Alzheimer patient brain, and which means that a myeloid cell, in this case a microglia cell, has been trained by evolution of 1 million of years and equipped with pattern recognition receptors on its surface to recognize these structures, and has been, learning that beta sheet structure is a pathogen. So they may misread the deposited beta sheet structure as pathogen invasion, and that then makes them mounting a very strong inflammatory reaction. Initially, this aims at, killing the opponent, eating it up, and then resolving the inflammation. And so I think this initial reaction is even a protective reaction, The immune system tries to battle the enemy and gets it away. Now, there is deposition which is, not a pathological and infectious challenge, it's a sterile type of inflammation, which is different. So there is no opponent to kill, which means all these inflammatory mediators will not hit a bacterium, it will hit the surrounding cells, damaging or impairing the surrounding cells in function, including neurons. So we know from some of the cytokines being released during this phase that they actively suppress, for example, synaptic plasticity. Now, this is one point. The other point is that the important part seems when this inflammation becomes chronic. So from this very immediate and maybe even resolving stage, becoming into a chronic active phase, where it may not be as flourishing, but it may be chronic and releasing lower amounts of cytokines, inflammatory mediators, but for very prolonged periods of time. And I think that the chronic exposure may be even more damaging as this initial insult, which tries to clear the pathogen away. So if you look at various factors, it seems like the initial stage of inflammation even protects, while the later stages might be the detrimental part.
It's always this kind of trait, protection and damage, and where this protection will turn into damage, and that is kind of, as you said, continuously there and harms our brains, unfortunately. We have neurons, glia, genes, cells, cytokines, different environmental exposure. How do you approach studying Alzheimer's disease?
I think there's no single approach, and it depends very much on the precise question you have. You have the subcellular level, maybe even the atomic level. Then you have the cell culture level. You can go to organoids or brain slices and to the whole animal. And then in my lab, it's always very important to look in human samples whether we find evidence that the systems we have been studying are actually important, and we find traces or evidence that they play a role in human pathology as well. So we try to go this ladder up and down. We either take a basic hypothesis, which we go from test tube to cell culture to animal model to even human tissue, CSF, plasma, brain tissue, and hopefully one day to the clinical trial, or we take a clinical observation and track down its origin in the opposite direction. But I think we have to walk this staircase up and down until we really dissect out the underpinnings and the sequential events that lead to the pathology.
And it's very complex. For people who are not familiar with this kind of business, there's a lot of companies that actually drop their research on neurodegeneration because it's just so complex and so hard, and there is no kind of positive outcome, unfortunately, or not to the extent that we wish. Or maybe correct me here.
I think we learned a lot from the trials from the last 3 decades. I also think we now have the advent of the first drugs on the market, also in Europe. I think from beginning of September, lecanemab will be on the German market and in some other European countries as well. It's on the market. We know that these drugs are no great drugs. They delay the progression of the disease by a certain period of time. We have to learn whether the individual patient senses this delay and improvement. It comes with quite some risk of intracranial changes, including edema and even bleedings. So these drugs mean we have to monitor the patients very, very closely by MRI, and I'm not quite sure whether we have the MRI capacities to do so with a great number of patients that would demand in future such a therapy. I think this is just the first step into the field of disease modification, and we have to learn whether other drugs and therapeutic strategies in combination with anti-A-beta vaccination are more successful, or whether we learn on one stage that we have another site for intervention, which becomes, so great that we do not need A-beta vaccination anymore. But in general, I think it's a start, and it's good for the field and it's good for the patients, even if everyone is aware that these drugs are no fantastic game changers.
When you say vaccination, this lecanemab is monoclonal antibodies, correct?
It's a passive vaccination, so it's the administration of monoclonal anti-A-beta antibodies.
I just wanted to make this distinction because now
it's no active immunization.
Exactly. Now it's a hot topic, or it has been for the last couple of years. These vaccinations, RNA vaccines, etc. And I just wanted to clarify this term. Given your expertise, what do you think would be a meaningful addition to let's say this particular monoclonal antibody that targets A-beta in order to reduce the inflammation, for example?
That's a good question. So I think an anti-inflammatory strategy would make sense given the evidence that, for example, risk variants drive the disease mainly through their activities in microglia and not in neurons or astrocytes. As evidenced by Kozo and colleagues in 22 in their very elegant Nature Genetics paper. But in general, I have an oversimplified model for that disease. Just believe it's a relay race. So one runner gives the step to the next runner. And let's call the first runner amyloid beta, the second runner innate immunity, the third runner maybe mitochondrial or metabolic breakdown, and the last runner is tau pathology. So one of the reasons that one therapy alone, like anti-beta vaccination, may not be successful is that if the first runner has made its round through the stadium and given the step to the second runner, you can break his leg. It will not change the outcome of the race, right? Now, the other interesting part is in one competition you have not only one team running, right? You have 8 teams. So let's call one entorhinal cortex, one frontal cortex, parietal cortex, midbrain, cerebellum, thalamus. So all these teams run at different speed. The moment a patient comes to me and reports his first memory deficits, the race is at full speed and it's ongoing. And it's like a visitor coming into the stadium and saying that the race has already started. So he will see one very advanced team who is already out with the last runner. He will see one runner which is very slow and maybe has not even finished the first round. And so it's a very heterogeneous picture, right? Which means we may, at a given time point, not be able to treat the disease with a single drug. We may have to have a drug that addresses at least 4 or even more of these mechanisms. And if I, would have to put my money on the best choice for something that modifies innate immunity, at the moment I would put it on the inflammasomes, on the NALP3 inflammasome, but maybe also on other inflammasomes, which are key innate immune signaling pathways, which have been shown, at least in the mouse model, to massively influence disease outcomes, and which also have shown evidence for at least being activated in human AD. And knowing that the pathway is highly conserved between mouse and man makes me hopeful that, modifying this pathway would most likely also benefit the patients. The key question is, when is the best therapeutic window? And so I believe we have to learn a little bit more about the sequence of events and when inflammasome activation, NALP3 inflammasome activation, is actually having an influencing role. Now, knowing that you have different brain regions in different stages of disease may actually indicate that you have a wider therapeutic window than we initially were thinking. So, but it depends that at a given time point, we may not protect all the brain function. We may only be able to protect a certain brain function. And we have to learn in which patients, which of these drugs are most effectively.
First of all, this was one of the best explanation that I've ever heard of the progression of the disease. Really, thank you about it. I've listened to so many talks on Alzheimer's disease, really with a lot of prominent researchers, and this was so incredible because it captures like so many details and shows you how complex the disease progression is. And maybe just because of these different stages and different brain regions, this is why we would potentially need some personalized medicine, but I will leave that out. When you said this analogy with the one runner giving the Staffel, I don't know how we call it.
Staffel, relay race.
Exactly. To the next one, I was thinking, maybe you are referring to neurons that actually spread, or amyloid plaques that are spreading around from one region.
I'm referring to mechanisms.
Yes. Yeah. But this is also something that in this disease, as you mentioned, you have different brain regions that are affected. And the disease onset is in, let's say, one region, it's kind of progressed through the whole brain eventually, and you have damage all over the brain. So it is just astonishing how complex the whole situation is, and I'm really admiring people like you and others that are putting so much effort to try and develop therapies to help actually humanity. I wanted to ask you about one aspect of your research, about this very interesting channeling nanotubes and microglia. What are these
Tunneling
things?
nanotubes.
Tunneling nanotubes, yeah.
So you know that cells are communicating, and cells use different, ways. So humoral factors, extracellular vesicles, but also tunneling nanotubes. And tunneling nanotubes are very direct lines. They're tubes which connect the cytosol of two cells, and they exchange messages, and they exchange cargo, and they exchange even organelles. So they can exchange mitochondria, they can exchange ribosomes, maybe other organelles as well. They can exchange single proteins, and that's a very direct way of communicating with each other. And we found that cells that we started with immune cells. We looked at microglia cells, and actually it was a project that had nothing to do and was not aiming at tunneling nanotubes at all. It was my postdoc Hannah Scheiblich, who had a very open eye and found these connections. And so initially her project was actually identifying to which immune surface receptor alpha-synuclein would bind to and drive phagocytic clearance. And then we saw that actually the microglia cell would overeat and accumulate alpha-synuclein. In contrast to beta amyloid, which these cells can break down pretty easily, alpha-synuclein would not be easily broken down. And these cells started to, distribute cargo to neighboring cells. They would call virtually for help and form tunneling nanotubes, distribute the cargo into these cells in order to get rid of what they couldn't, if you wish, digest. And then the neighboring cells helped breaking down the alpha-synuclein aggregates. Interestingly, while they received the cargo, they delivered fresh mitochondria to this microglia in need, and this microglia cell then survived. We showed that if you would compromise the delivery of mitochondria, that would actually also compromise microglia survival. So it's a metabolic, it's not so surprising, it's a metabolic question, so to say. And then I remembered a picture which I have seen a long time ago, because a friend of mine, Helmut Kettenmann, has shown me the original of Alzheimer's publication, and there was one cell which Alzheimer has denoted as amyloid glial cell, which sits on the surface of a neuron, and it contained particles which were colored by Alzheimer the same color than the neuron. I thought, God, when they do not only communicate via themselves, maybe they also communicate through tunneling nanotubes with neurons. And indeed they do. So they're tunneling nanotubes between microglia and neurons. And we have great evidence that they really free the neuron from alpha-synuclein and from tau aggregates. preventing dysfunction and death of these neurons. At the same time, they also deliver mitochondria, which I find totally exciting. So when we stopped or when we intoxicated the mitochondria in microglia, then we would drive neuronal death. So they would not get the necessary metabolic help through that mitochondrial delivery. If we would allow the microglia to deliver fresh and functioning mitochondria, that would rescue the neuron in function and survival. So I think that's totally exciting because, maybe we could train our microglia to do that more efficiently. And now we have to look, how does that look in disease circumstances? We have actually for quite a while studied this in vitro, and it has been always questioned whether this is a mechanism that occurs in vivo as well. But very recently we were able to show in the zebrafish model that because the zebrafish is more transparent and it's easier to do a live microscopy in vivo, so we found that the very same mechanisms are in place in the zebrafish brain as well. So I strongly believe that we can use that maybe as a future therapeutic intervention by training the cells to do that in a more efficient way.
I have so many questions to this. By the way, I did my bachelor's thesis in Professor Kettenmann's lab. So
very good.
That's why I love microglia and glia in general. And when you said that, Aldous, he showed you this image of back then. I at some point during my PhD, I started to think like, there is nothing new since the beginning of 1900 that we discovered. These guys, they drew everything by hand and they saw so many details, Cahau, Golgi, Alzheimer, and Virchow, if you wish. So it's just incredible the amount of knowledge that they provided us. And now we can basically build the tools to confirm, we have here microglia, we have astrocytes, these are oligodendrocytes, it's just incredible. But as I said, I have many questions to the things that you just said. It's incredible in so many levels. First of all, that mitochondria, they also have their own DNA. And is it different, the microglial mitochondria than the neuronal, for example? How does this influence the different cells? Also, just one clarification that you mentioned alpha-synuclein, and this is another protein that is important in another neurodegenerative disease, is Parkinson's disease. Very, very important, just to again clarify it.
And Lewy body disease.
Lewy body disease, exactly.
As a cortical phenomenon in cortical neurons.
Exactly. And we had 2 episodes back, episodes on memory and how some memory processes are actually happening also in other cells, not only in neurons, like these kind of cascades, pathways that are canonical for memory formation. And this is cool to when you say we have to train microglia. So if they kind of remember this and we can train them to provide neurons with more functional mitochondria, this is a great maybe therapy in addition to the monoclonal antibodies or whatever is out there. But when they attract other microglia, don't they also spread to these cells the pathology?
Eventually. It's a good point, because you could say, well, the recipient microglia may spread it then even further, and sometimes we really see that if one microglia that receives cargo, it even gives it to through the next one. So I think it's all about the efficacy of clearing mechanisms and you see in the GWAS hits and from other evidences that lysosomal processes are really important. And so I think as long as we keep our waste bins functioning, then and these degrading proteasomal or lysosomal or autophagosomal machinery going, that will be effective. In case it's then not it's not effective, it may be passed on to a variety of other cells, and maybe not between neurons. It's interesting if you think about transsynaptic spread Frank Kirchhoff, who actually worked here in Göttingen, showed me an electron microscopy, I think decades ago, where we showed these tiny microglia processes scanning the synaptic cleft. And you could imagine that as long as your microglia cell is active, attent, and so to say awake, it would sense these particles and take them out of business. If the microglia cell is compromised by immune senescence, maybe on the basis of certain genes, risk variant, for example, that system could be compromised and then spread much faster. And there are models where you can study spread of pathology in the context of Parkinson's disease along the gut-brain axis. If you compromise these models by a systemic inflammatory challenge, the spread is much faster. So I think there is evidence that there is a interaction with the immune system on many levels. In the disease, we just have to define them further and see the sweet spots where we can interfere and do something good for our patients.
Professor Heneka, I would love to talk to you for another 1 hour or even more, but we have to bring you back to the conference. And I'd like to ask you one last question. As a person who is in the field for many years, you've seen patients, you've talked to prominent researchers. What kind of advice would you give to people, let's say middle-aged, young people even, to try and prevent, although they don't know if they are going to get any disease in the future, but just try to do their best to not get it?
Eat healthy, work out, keep curious, put yourself on a higher education level. This is protective for sure. And then you already do a good thing. If you're actively doing sports, then you don't become obese, you don't become hypertensive, which is a risk per se. I think you can do a lot with sports, continuous movement, endurance training, and good diet. And may not be so popular, but alcohol is also a risk factor, and binge drinking may not be good for the brain after all. So maybe it would be good advice to, if you drink modestly.
And also sleep good, guys.
And sleep good, yes. Sleep is important, absolutely.
Very important. Professor Heneka, thank you very much for your time and for your insights on Alzheimer's.
Thanks so much. It was a great pleasure talking to you.